We're glad to be back here in real time all of a sudden. And a couple of things. Oh, let me put it over here. So I do not know, and certainly if you had a chance to listen to the recording, or if you didn't have a chance to listen to the recording, either way, it isn't a big deal. I used this Nestor Anderson a fair amount. I used both these PowerPoint sequences. I meant to put it over here, but it's under this week one outline for whatever reason. Still, Ultra's a bit of a pain when you put these things in. So you can certainly follow along with this. We're going to be flip-flopping between those two PowerPoints in addition. So keep an eye on it. And I'll try to put the package all together. Surely because of today, and surely by Friday, everything will be done with this particular first chapter. And then we really move on to I just like to do the background thoroughly. And today's really important. So what I started, and maybe I'll go back and just do a mini summary while I'm thinking about it here, with what I call, it's Anderson, but it's really Nestor's Microbiology is really, Anderson was the most recent. And I went over a lot of these particular things. If you didn't get a chance to listen to it, just a quick summary is what I'm doing here for all of you. And we see, and I didn't get that terribly far. I really wanted to look at the historical figures. So I talked a little bit about, and to me, that's more lab related stuff, how the microscope got, but to understand, I try to give you the background of really the crux of today's discussion. Really, that's what I framed it. They didn't know, they thought there was some miraculous force in the air that could make this, eraser come to life. Evil forces in the air. Science and medicine down right after the fall of the Roman Empire. We called it the Dark Ages for reason. Well, it's not an indictment of the church dogma, but that, to a certain extent, that's what it was. I mean, they couldn't accept that there were things that you couldn't see. There was disease and illness and injuries and damage. It just was frowned on. Oh, it was God, the devil, all that type of stuff. Something, some terrible evil force in the air. So, and the idea of these things just happening with spontaneous generation. I mean, that was the state of the knowledge, and you couldn't go against church dogma or anything like this. And I presented this to you. And another term that we use a lot is the A. Remember, A means without, it's all the word. Biogenesis. And the whole idea, going through these historical figures, starting with the Renaissance, they began to question it because they were scientists. They made observations. They observed things and they started to test them. And so, I mean, this was early on. I mean, you're going to see these under the microscope. I don't know if I realize it's a bad week to ask about all this going on. But, I mean, I know but did you start to look at the microscope in lab yesterday? You see these guys. Cocci, bacilli, that's with a small b, not with a big B, we'll get there. Larger bacilli, spiral forms called spirilla. And you're going to see the impact of spirochetes when we get to them very, very famously. Think Lyme disease is a spirochete. So, I mean, that's where we marry the two, lab and lecture, together. So these things they just needed something to see them, hence the microscope, and to begin to understand them. And so we went through these. There is actually the original microscope. We used to make those things in college. This was that idea, and it took years, starting with the early Renaissance. And believe me, it took the better part of 250 years. It started with this guy. We probably started before, but Francesco Redi was able to, and if you listen to that YouTube lecture that I did, okay, basically, if it was 3 jars, not 2, he made the scientific observation that when flies lit on meat that was left out, okay, shortly thereafter, you start worms appear. And there's a force in the air that makes worms grow out of meat. Okay, really? Well, he figured, hey, the flies are laying eggs and worms grew. And so he did a very simple experiment. He had one, it was basically a glass petri dish. We use plastic ones in the lab. We have glass ones. And he put some meat in 3 of them, not 2. One, he just left the way it is. Flies lit on it. There come the maggots. Okay. He put a glass cover on the other one. Well, nothing grew in that And that was the problem because he wasn't letting the evil force in the air, get to it. And he covered the other one, he's Italian, with cheesecloth. So, I mean, and if you're familiar with cheesecloth, I mean, cheesecloth comes in different thicknesses. You put multiple layers, reasoning that even if the flies got on it, the air could get to it, but the eggs wouldn't get to it. And he was absolutely right because maggots didn't grow. Pretty simple experiment. But the whole idea, oh, the cloth pushed away the evil spirits. Oh, this wasn't accurate. And people couldn't duplicate them. The whole idea is called validity. And that's really an important point when you try to validate an experiment. You do the experiment, I can do the exact same experiment halfway across the world or in the next county or in the next university. And I should be able to get the same results. That makes them valid. And that's what scientists do. We're inherently skeptics. And maybe you didn't like them, and they're publishing a paper, and I'm gonna show that this really didn't work. I mean, there's a lot of times it's a positive attitude, sometimes it's a negative attitude. Be very fair. So that's all part of the backstory from this. And so other people were starting to get this idea, and what they used a lot early days are things you're using in lab. I assume it was Dr. Dress, did he go over tubes of broth and plates of agar? We use broth all the time. It's a nutrient media. That broth is interesting stuff that you mostly use. It's a mixture of typically pork or beef extracts, okay? And they have this thing called peptones and a variety of things. So basically it's a soy extract, which is a protein, which is a complete protein. I want to go back over that ground again. I don't want to go back to AP1 again and me doing my song and dance about complete proteins. But I'm willing to do it. I broke down yesterday. I have a confession to make. We were out shoveling. I actually got out the long johns. It was cold. That was cold. Just saying. I have to confess. Nevertheless, they started to working with broths, and they were trying to see if they left them open in the air, and they had different kinds of flasks and sealed the flask. Well, the seal of the flask took away the air and blah, 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 until it was cracked. And so heating destroyed the vital force. Bunch of baloney. So it's all about Pasteur, which leads us to the single most important discovery. And Pasteur's research was faulty. He knew what he was doing. He had credibility. He basically developed a method today we call pasteurization. We'll go into it more because there's variations of it. And nearly anything that you consume or I consume is pasteurizer. Okay? Because it basically, effectively what it does, your dishwasher. Basically what it does is it is able to destroy microorganisms, typical kinds of bacteria that are most likely to cause, intestinal problems. That's really what pasteurization does. It's not the same as sterilization. Okay. He experimented. His credibility came from the wine industry. People in the wine industry knew he was a brilliant scientist, knew he looked at things through the microscope, and what he was able to ascertain in an effort was what was causing wine was spoiling at an alarming rate, and it was a big industry. And so he developed this method to stop the spoilage of wine. So they made the wine, if you didn't drink it right away, and had it in whatever barrels, however it was stored, or bottles, however they stored it, it spoiled relatively quickly. The reality was that the yeast that were fermenting it, continuing to ferment, and what they do is they made the transition from alcohol when they continue to ferment, okay, to vinegar. They named it vinegar. I did this in talk. Vin agra, vin wine, agra sour. From the Latin. That's where you get the term vinegar. So if the wine goes bad, it's vinegar, which is not. We use it intentionally. We have red wine vinegar, white wine vinegar. It's a good thing for cooking, not a good thing for drinking, as opposed to having a nice glass of wine. So like for red or innocuous alcohol. Nevertheless, he experimented with temperatures and time, and he found that if he heated it roughly to 140 degrees Fahrenheit, 62.5 roughly, 62 to 63 degrees Celsius for 30 minutes, numbers you will need to remember going forward. And it didn't eliminate, if you boiled the wine, you eliminated the taste, it kind of destroyed it. So this maintained the food quality, if you will, and at the same time, when it cooled, it didn't spoil. So immediately he was credible. And what happened was he had looked at things he found in the microscope, things he found that were in the wine. Similar, he was seeing yeasts, other and similar yeast as fungi and bacteria that was something that he was familiar with, and he saw that in the wine. So he knew, he came up with what he called germ theory. And germ theory was just that. Tiny microorganisms that we can't see, and he called them germs, just like we do today. And germs is a garbage term. Basically, it's not a very, includes all microbes, so to speak. So his idea with germ theory was things we can't see was directly opposed. Directly opposed, okay, to this concept of spontaneous generation. He refuted it. How did he do that? He began to look, doing the same thing. So he observed microorganisms, and I'll show you, and I commented on it a little bit. I really stopped before I really wanted to get to the crux of the matter. He had the glass blowers make these flasks, and he put a broth in. Now, he used a sugar yeast broth. This was the error. And he put the broth in these flasks, and what happens, he had the glass blowers make this sort of a goose or a swan neck flask. And his idea was, I didn't mean to do this the second time I did that. His idea was, as I boil it, all the stuff in the air and these little tiny microbes you can't see were going to get pushed up, trapped in there, because he had done it experimentally. He had some cotton in there, and he took a look, and he found from slicing the cotton and looking under the microscope, there's a whole bunch of stuff I've seen before. And he did that, and it worked. I mean, he had some of these flasks for years that stayed sterile, and they were open to the air. That was the idea. Well, the air can get in there, and it's not damaging them, and all I had to do was tip it or break the top of the flask or anything like that, and within a relatively short period of time, and wait till you see how fast things grow in the lab, in a matter of hours or days, if you came back in 12 hours, it would be murky or cloudy. The term we use in the lab is turbidity. We can even measure it. And that's something that's done in the laboratory when you're doing standardizations and things like that. I'll leave that to the lab. But when it gets murky or cloudy, it ain't sterile. When it's a solution, when it's clear, it's sterile. Everything was right about what he did, but people could not duplicate it. They couldn't validate his work. And so skeptics had an easy platform to say, hey, Pasteur's walking out of you know where. Okay? The whole idea was the difference in the broth. He used a yeast sugar broth. Yeast sugars were easy to boil and to sterilize. The other people didn't use the identical broth. That's the whole idea when you do experiments. You do things the same exact methods and materials. The whole thing we write a lab report against methods and materials. A lot of people used hay and fusion. Fusion is just a generic term. They would pick hay that wasn't off the ground, and they would cook that, and they would basically use that as the framework and put water in it, heat it up, and effectively that was called a hay infusion. They used a lot of those in early science. The problem was you're taking it off the ground. It wasn't just the hay as a plant source and getting nutrients out of that the way you and I might make a cup of tea. Okay. Soil was a player. And so this was where the problem was, and it's why experiments have to be exact duplicates to validate them. So in the same year, a German botanist named Kohn discovered endosperms, heat-resistant forms of bacteria. All Pasteur did was boil them. He actually heated them more than he did with pasteurization. They were extremely heat-resistant, and this was the distinction between Pasteur's results and the others. Pasteur used broth with sugar or yeast, highlights the importance of reproducing, which I'm blabbing about, all the things in conducting research. The trouble was the soil. The soil has stuff in it. Called endospores. These are tiny things you can see under the microscope, and you will see them and you will stain for them. That aren't destroyed by boiling. Boiling is not a substitute for sterilization. It's not. When we sterilize things, and we do a whole unit on that, we'll talk about pasteurization. When we sterilize things, we don't heat them to boiling, 100 degrees Celsius or 212 degrees Fahrenheit. We superheat them in a pressure cooker. Okay? You got an Instant Pot at home, you can sterilize. Basically, all of this a little bit different than that, but more or less that's what's true. So they found things in the soil, and you already know about that. We are constantly on guard for these things. Constantly. When someone comes in with a wound in the hospital, what are the questions they ask them? How did you get the wound? What medications you're on? Allergic to any medication? Now, what's another question they ask? Something they always ask when you have a cut or a wound or anything else. It has to do with something you had. When did you last have it? God bless you. Tetanus shot. Usually I just pull out my wallet and try to be funny, and I go, what's the important thing they ask you about is that? The health insurance. Who's paying? I figure you guys the stupid jokes, because you've heard most of them. Why would they ask you about tetanus shots? Well, frankly, the reason they ask you is for the next the time, not when you're in the hospital or the emergency room or urgent care. They're asking you for when, heaven forbid, something bad happens. There's an accident, you can't get treated. Honestly, with regard to that. Why? Tetanus is everywhere. The name of tetanus is a bacterial infection. It's called Clostridium complex. It's capitalized. Genus. This is called genus. The species we always make small. And it's an endosperm. Because in a wound, the wound is not cared for properly and begins to necrose. Fancy word, tissue dying. The tetanus will flourish. Tetanus grows in an area or in an environment, better word, without oxygen. So we call it anaerobic. It means it thrives without oxygen. And it's made for, and the reason it's always there is endospores are indestructible. We'll go into this in the next chapter. They live for not a year or 2 years. They probably live for a couple of 100,000 years or more. And they're in this carpet. They're in all of your shoes. They're in your environment. They're in your dorm room. They're in your house. They're there. We carry them in. We do this all the time. Horrible freezing weather we're having. Endospores. Hey, bring it on. We have to use extreme heat or extreme methods of work. Clostridium tetanus. That's what we're concerned about tetanus. Also, you get 3 shots in one. Tetanus shots last for about 10 years. And you get diphtheria immunization, and you also get pertussis, which we'll go into all those diseases over time. So you get, the key is the tetanus, because the problem is, if it's a really nasty, what we call a tetanus-prone wound, there's significant. I'll get into that down the road. Not done yet. This just shows you how bad endospores are. Next endospore. Let's see, and the name is important. Food poisoning is generally not fatal. I mean, if somebody's very, very ill or very at risk, food poisoning, vomiting, and diarrhea, but usually it's from cooked food that's been cooked improperly and it develops toxins. And most of us have experienced it one time or another. Well, there's one kind of food poisoning that is potentially immediately lethal. It's a very interesting toxin. We take teeny tiny amounts of this toxin we talked about. Last term, we talked about exocytosis. Okay? And we take teeny tiny amounts, micro amounts of this toxin, refine it, and actually use it therapeutically, cosmetically, for toxin. Wrinkles. Botox. Here's its name. Clostridium botulinum. It's true because they're all in the same genus. It's also an anaerobe, and it thrives in anaerobic environments. We don't usually see those, but in food preparation, like I mean, if you're one of those folks, and we do a lot of that stuff, who grows stuff in the garden and wants to put it, and basically preserves it, cold packs it, which means you heat it up, put them in those mason jars with the lids, right? Okay, you gotta know what you're doing with that. Somebody likes to do, when they tell you, there's somebody who likes to do, you know, your own beer and that beer type things, you've got to be very cautious with sterilizing. It's very easy to get. There's a food where we do see botulism, and there's been some problems recently. The most common food that we have to be very careful when we give it to infants, their immune system development, contains botulism potentially is good. Very good. I think I told you guys this. I had a student came in from home who was making meat, honey baby. And there we tested it. It was botulism. So the endospore test, flu botulism, and the classic wounds. Tetanus isn't the only bad word. We don't see it so much now with chopper evac. It was Civil War, World War I, World War II. When you had big explosions of armaments and things like that, you had bone exposed to the air, horrible wounds. What you worried about tremendously was a disease called gangrene. It would become gangrenous. And it was extraordinary because your only alternative then, if you got to it soon enough, was the name of the organism that typically causes that. Clostridium perfringens. That's right, another endospore. It's in the soil. Interestingly enough, when we get to antibiotics, we're going to talk about sulfa drugs and the fact that Allied troops in World War II had a drug called sulfanilamide. We'll get to it. A powder, they would sprinkle it on the wounds. Hurt like hell. But it was very effective at limiting gangrene. Again, wars are about attrition. I'm a World War II buff. The wars are about attrition. And so one thing, we had more troops, and with the Axis forces, the Nazis, the Japanese, and the Italians didn't have that. So when they got war wounds, and what they did, they ended up with amputations and everything. War of attrition. Make no mistake, World War II was one of them, was attrition. And a lot of it had to do with antibiotics and that moving forward. Clostridium perfringens. So we're very, very careful. If you're going to go in, if you're going to emergency room work, if you're going into healthcare, you're going to see this. Very careful. The one you're going to see the most of is in the same family. And many of you, if you're working on a clinical floor, will be able to walk down the hall when you get to the room before you get in there. You'll know this disease. By smell. By many of you are working, maybe you are volunteering in healthcare facilities, nursing homes. What's the name? Remember, when we write these things in, we don't have to write Clostridium tetani, we can just write C. Something that starts with a C. C. diff. See, you're interactive. It means Clostridium difficile. Difficile because it's so darn difficult to deal with. And we've nicknamed it C. diff. There's treatment for it. We'll get into that type of a thing. C. diff is normally in most of our guts anyway. We take strong antibiotics, it kills the normal bacteria in there, and the endospore can flourish. And the inside of your large intestine, everybody, is an anaerobic environment. Check, please. That's the interesting part. And because Pasteur, getting back to him, didn't use the hay infusion, what? Didn't deal with the endospores. And that's where the problems came from. Okay? The importance of doing it. And so Cohn was a very interesting, he was a botanist, okay? And as soon and the spontaneous generation was disproved, the golden age of microbiology was born. The principle of microbiology causes disease is called the germ theory of disease. We didn't get to Coke. And this is an interesting chart if you want to look at it. I'm not going to go over it. Fascinating. Which brings us to this. Heat-resistant microbes, labs that could not reproduce, failed to use broths made from hay. Which brings us to the distinction between Coke and Pasteur, and Coke was younger than Pasteur and lived longer than Pasteur. There's a reason Pasteur did not receive a Nobel Prize. Pasteur died. Surely it was worthy of it. Pasteur died before the Nobel Prizes were started. They do not award them posthumously. The only time someone who's dead gets a Nobel Prize is if they died after they were awarded the prize, but they still get a ceremony. Died right after the award. And that's happened. Pasteur, in addition to pasteurization, in addition to germ theory, was the first one to coin the term virus. He was the first one to develop a treatment for viruses, particularly for rabies. Pasteur was all about treating people. It was all about finding solutions to people's problems. So he was much more clinically oriented in that regard. His friendly rival, Pasteur French, who was Koch, who was German. Koch did get a Nobel Prize because he was a little bit younger, and then probably, I think, 1906, something like that. He was the ultimate lab geek. He did everything in the laboratory. He was the first one to successfully grow endospores, and he all the techniques you're going to do in lab, the agar that you use came from Koch. The way that you played out cultures, Koch. Growing, his whole premise, you've got to grow things in pure culture. And if you do that, he developed something that we'll talk about more than once called Koch's postulate. It was like a chain of evidence about how diseases are caused. What he did, and there's a lot of illustrations, you'll see it in some subsequent chapters. The disease he was dealing with, and I wrote there, is called anthrax. It's caused by an endospore. Why it was much more commonly seen, and it was a problem, this endospore anthrax called bacillus. Notice the big B. When you use a little b, it means that rod-shaped bacteria. When you use the big B, okay, now you're talking about a genus. And anthrax is aerobic. It's in our environment all the time. And anthrax was a disease that was affecting a lot of sheep, particularly goats, cows. It was very common, and it was affecting the people who work with them. Okay. And so he was able to identify this because it would grow easily in culture because it was an aerobic, and you're going to use, not anthrax, obviously. Anthrax could be a bioweapon. It's been weaponized before. But we are going to use one called Bacillus subtilis, which we finally call B-sub in the lab. And what's neat about that is we actually get antibiotics from that. That's where we get something called bacitracin, which is part of triple antibiotic. And we'll talk about that. Neosporin, polymyxin B, and bacitracin were the original triple antibiotic that was made by a laboratory developed right here, and you guessed it, Pittsburgh. I don't know why the North Jersey, New York East guys are so fond of talking about that, but it's true. Helps the lecture. So that was part of why he was so interested in. What he did was he had an animal that was dying by anthrax. He had sores. Aspirated, took a needle, and he pulled out some of the fluid from the sores, and he grew it. And it formed a particular growth on a plate in the lab. Then he extracted the fluid from where those colonies, which you can see, in the laboratory, and he injected them in a healthy animal, and they died of the same disease. Not good for the animal. Okay? And then he was able to extract the fluid from that dead animal and grow it again in culture. That was like, here I'm showing you exactly what caused. It was like cause and effect, and I can prove it. So for a long time, it was Koch's postulates. They don't apply today. We use similar postulates. We have a molecular basis. And for disease, we'll get to that when we talk about disease and causes. But understand, he was all about the laboratory. He identified tuberculosis, staphylococcus, streptococcus, nearly all of the bacteria that were common and that we utilize in the lab today, or we utilize common diseases, he was able to identify. And that's kind of where a lot of this discussion comes from. So, because of that, we have a handout. And so, this is why when I send this stuff to you, take a look at it. So, here's one. Pull that bad boy up. I know you can't see it with this. I hate this. Come on, Lou, you can do this. You don't have to do it. You're up. Okay, I can't expand it, but you can do that. I'm having trouble today. Oh, I can do it here. Look at me, stupid guy. So this was the summary here of the Clostridioms. You notice when you write them, you either do them, underline them if you're drawing them, or you use italicized if you're writing a report. You won't be doing that for me. Just a little hint. Okay, got it? Good. So that's a little bit on the historical figures. I have some of that. So I have another one of these handouts here and it's all on your blackboard for the domains, some of the things that we had talked about, how they replicate, binary fission, which we're going to go into. They don't do mitosis or meiosis because they don't have the same kind of anatomy. We'll get into all of that. That are there, okay? Okay, we're going to look at that in the subsequent chapters, all of these types of things. So we're going to look more deeply into all of these things there. But this is just a little quick mini review of that for you. And lastly, this other handout that I have is these additional notes. This is a little bit more about, I'll try to blow it up a little bit more if you can see it. Genetic engineering. So I commented on that a little bit. Basically, it utilizes either E. coli or yeasts because they're relatively simple and they're easy to grow in the lab. You have a tough time, believe me, in the lab. You have a tough time, particularly a typical micro lab in a college, university type lab, of not having contamination with things like E. coli and staphylococcus. They're everywhere and they grow like crazy. Okay, it's easy to get contaminated. So they're very easy for us to use. And we have a lot. And E. coli isn't necessary. E. coli is a very, very common gut bacteria. There are certain strains or types of E. coli that are nasty. Fortunately, not any of the ones you're going to use. Okay. There's all different classifications in the lab. We are what's called a modified BSL-1 lab, which means biological safety level 1. When you hear about terrible diseases like Ebola, and when they were originally doing the investigation of COVID-19, that was leveled as BSL-4, something that could cause immediately life-threatening disease. Walking around in those encounter suits and things like that, you get the idea. Modified BSL-1, these are agents that are bacteria that are, or organisms and other agents that are not known to cause disease, okay, in healthy individuals. We will have Staphylococcus aureus and a couple of others. It's a very tame strain. So unless people are immunosuppressed, and I always encourage my students in lab, if you have any kind of disease or entity or allergy, talk to your laboratory instructor, say, hey, like when we work with penicillin in the lab, and we do, because we do testing for antibiotics, my students would come, well, I'm allergic, so I use this. I had a simple answer for them. No. Why would you do something like that? You're all doing the same experiment. I'll look at somebody else's results. Okay. You understand what I'm saying? If there's an underlying condition that you have or an illness, let your instructor know. The same way you would let me know if there's a problem. I can't be here today other than it's a freaking deep freeze outside, which is fine. So we utilize this because they're easy to grow, and a lot of them are very receptive to genetic modifications, and we'll go into those when we look more about bacterial genetics. I talked about the plasmid, lots of interesting insulin and hepatitis B vaccine, human growth hormone is in this cluster. There's a lot of neat things. Oxytocin, we call pitocin. We'll get into all that stuff. Gene therapy, look at the viruses uses, HIV. Gene therapy is actually our resequencing cells. HIV we take the bad out of the HIV. We utilize an enzyme that comes with it called RNA reverse transcriptase. Basically, it's an RNA virus that rewrites itself as a DNA virus and it inserts into our DNA. So in order to change a cell or a cell line, you have to overwrite the DNA. The same way you might overwrite a computer program. Okay? The same way you might insert an app into using whatever computer analogy that's there and change something that you're doing. Okay? It's not a whole lot different than that. That's really in its early developmental stages. I comment when we talk about vaccines, I'll talk about mRNA vaccines. That's a little bit different because you're just making them basically instructing that cell to make a specific protein as opposed to basically resequencing the cell. Okay? That's an important topic. This last stuff is really important. This is the two types of diseases. One is called emergent, ones that are new to us, not necessarily new. Why would they be new to us and not necessarily new? Well, we go to places, we build everywhere. We're building in rainforests. We're going into climates that normally people don't live in abundance. When you're doing that, you encounter new germs, for lack of a better term. So emergent diseases are ones we're encountering because we've never been there. But more often than not, they are mutations. I told you, MRSA is a mutation. COVID-19 is effectively a mutation. Whether it came from an open air market where they had bunches of dead animals everywhere as food sources and living animals that they were going to butcher in a very unsanitary environment, or it got out of a lab either accidentally or by intent, nobody knows. I'm not being a conspiracy theorist. Nobody knows for sure. And there ain't nobody talking. But understand, those are all how emergent diseases. There's a ton of emergent diseases. I mean, HIV is effectively an emergent disease. There's a lot of good science that shows that was an effort to make polio vaccine in sub-Saharan Africa that was done in something, and I'll go over the story again in detail, in something they made a lab in the middle of the jungle with dirt floors, and they were growing it in monkeys, different kinds of monkeys. I don't know what the appropriate genus or species is or name for that particular monkey, but they were diseased animals to start with. When you start growing a virus in someone that already has a disease, perhaps another virus, they intermingle. Not unlike, okay, what brought us COVID-19 and related problems associated with it. So that's what we mean by emergent diseases. So genetic mutations, recently discovered areas, recently discovered environments where people are expanding and living. They're really important, okay? A resurgence is a disease that was near eradication and began reappearing in larger numbers. The best two examples in our area are tuberculosis and measles. I'll address them both. Typical causes, I'll move it in. Vaccine laxity. Vaccines are controversial. They probably should not be. I will frame it this way. And again, everybody, you're going to get my spin on things. You may talk to somebody else who has a different spin. I believe in vaccines, but they need to be safe. And there's no reason pharmaceutical companies cannot make a safe vaccine. They can't. It's all about the preservatives or the headaches that are associated with vaccines. And they have been for years. And that's another one of those things where nobody's talking. If you did not know it, the companies that make the vaccines are immune from lawsuits. There's a law that was enacted in the 1980s during the Reagan administration. I was a big fan of Ronnie, but that's the worst law he ever enacted. So basically, I'm trying to think of the right word, indemnify. Fancy word, meaning you can't get sued. So if you get sick and you can prove it was a vaccine, difficult to do, suing the manufacturer you can't do. And the doctor will say, well, hey, I bought it from the manufacturer. Go after them. They told me it was safe. Not going to spin it any more than that. But vaccine laxity, and we're seeing it. That's where measles came from. Measles is maybe the most highly communicable disease. Go back and look for measles outbreaks. You're going to see that all in this country. Measles is easily the most easily spread disease that we know. When I was a kid, 99% of us got measles. We all got it. Pardon, you gotta tell us. My mother, along with a whole other wonderful moms, okay? It's a quick call. My mom associated all these other moms. Yeah, at the end of first grade, they took us all to the circus in New York City at the old Madison Square Garden, which is 50th and 8th Avenue, no longer down there on 33rd and 34th and 5th Avenue, which is now an older arena. So we all went there. Oh, we're going to go eat at the Automat. Look up what an Automat is. Everybody was very, very excited. And all of us kids in it, because we were all the same. We were all people who had moved out of the city into a suburb 7 miles away. And they all had little kids. We were all great. I can't even tell you how many kids were at the bus stop between K through like 6th grade. It was immense. My sister was one of the oldest, and she was in 6th grade. So he took us all to the circus. As if by magic, we all came down with the measles, and then a few weeks later, we all came down with the chicken pox. Moms then used to have chicken pox parties and measles parties. They wanted you to get them so you could get over them, and we all got them. Child abuse. Where's that 1-800 for a child? See, Mom, you got off because we don't have technology. Absolutely true. Ask your parents, or in your case, ask your grandparents, okay, if that happened to them. We all got that. We were called the usual childhood diseases: measles, mumps, chicken pox, German measles. We're going to go over all of those. Okay. The vaccine, the MMR, measles, mumps, rubella, which is German measles, from the time that came out, basically it didn't happen. Chicken pox was later. All three of my kids, and they're right now roughly between ages 40 and 43, all got the chicken pox. But they didn't get any of the others. Very effective vaccine. I mean, it's crazy not to get that vaccine. Very, very little problems with it. Very safe. But people, because of some, this is not spin. Somebody put out an article indicating a very, very bad preservative and did some very, very faulty research that was published in a prominent British medical journal. And we're going to go over it. It had to do with a mercuric compound called thimerosal, and implicated that in autism. The research was terribly faulty. It was withdrawn. The publication, prominent British medical journal, maybe it was the Lancet, said, we're sorry, we messed up. This guy did terrible research. But the whole people against vaccines went nuts. And the reality is there's been an increase in autism, and the reality is harsh preservatives are used in vaccines. And unfortunately, that caused a lot of people to stay away from vaccines that were proven over the test of time. And that's why you're seeing some of the changes. And I realize a lot of it, it's been politicized, and medicine should never be politicized. It just makes no sense. I mean, yesterday, arguably, the top 2 football coaches in NFL history, one of them didn't get into the Hall of Fame on the first ballot. Bill Belichick. He only had 6 Super Bowls. Sorry. Politicized. Because people didn't like him. He's a pain in the ass. The whole sports world is going nuts over that. And they should. I mean, that would be like if Tom Brady doesn't get into the Hall of Fame on the first ballot. What are you talking about? Politics are everywhere. So vaccine laxity, worldwide travel. I don't know about you. Our big trip was driving 50 miles to the Jersey Shore. Okay. That's as far as we ever went. I didn't go on a vacation with my parents, and I didn't go on a vacation until I was like, 14. Didn't have enough money. And not bitching. I certainly wasn't starving. Just didn't have a lot of money. But now, all over the place. Okay, we're going to have people, and maybe you're one of them, and God bless you if you are, that you're going to go away. Oh, we're going to the islands. We're going to Cancun over spring break. Okay. Well, it's nothing to be traveling all over the world. My son's going to be living in Bahrain for 2 years, and my daughter-in-law and my 2 grandchildren on that side. Oh, you're going to come to visit us? Really? Okay. Worldwide travel is common and spreads things easily. Aging population. We are living, thank goodness for this one, much older. Very proud of my wife and I for digging out our very large driveway by ourselves. Okay. We were out there, not stupidly. I'm out for 15 minutes. Oh, it's freezing cold. I'm going back in. But it's good. Pretty happy we were able to do that for ourselves. We're in good shape. So people are living longer, and unfortunately, diseases can come back. And I'm almost done blabbing. And then we have so many we have people for transplants, we have immunotherapy. It's everywhere, every freaking TV commercial. And what has happened? Tuberculosis, which we had nearly eradicated without a vaccine in this country by just doing, by just doing chest x-rays, TB testing, and treatment when necessary. And we nearly had it completely eradicated. It came back. Part of it had to do with immunological diseases like HIV/AIDS. Part of it had to do with all of these other factors. Okay. Tuberculosis is a very interesting disease. The major cause of tuberculosis is overcrowding. That's why it's rampant in India. We do not have a vaccine because if you use the vaccine, you are not able to use the TB test. You always test positive. You will see if you travel to certain countries, India in particular, where TB is rampant, you have to get that vaccine in order to go there. And it doesn't work in adults anyway. But in order to get a visa to go there for whether you want to visit and you want to see the Taj Mahal, or you're going there on a business trip or a medical mission, you're getting that shot if you're going. So all that. My daughter had that. My daughter, if you didn't, I think I told you, she was there at least 3 or 4 times doing egg donations. More on that down the road. I'll leave you with that. Thank you very much for allowing me to drone on. I know I'm droning.
Yeah.
Still one of my favorite ones to do.